Hereby I open this academic ceremony in which Mr. Berger will defend the academic thesis Cryo-electron tomography in FIB-lamellae for the structural characterization of bacterial secretion systems. I welcome you, of course, and wish you lots of luck. And I welcome also the supervisors, Professor Peters, Professor Ravelli, and Dr. Iglesias, and of course the members of the opposition whom you will meet, and of course all people here present online and on site. But before we start the opposition, I give you the floor for a 15-minute lecture about your work.
Thank you. Dear Pro-Rector, members of the Corona, colleagues, friends and families. In the coming 15 minutes, I will give you a brief overview of my thesis titled Cryo-electron tomography on FIB-lamellae for the structural characterization of bacterial secretion systems. So I'll first explain what are bacterial secretion systems. So bacterial secretion systems are protein complexes that bacteria use to infect host cells and hosts, like you and me. In this thesis, we try to structurally characterize the type 3 and type 7 secretion system. The type 3 secretion system is structurally relatively well known, whereas the type 7 secretion system at the start of the thesis project was mostly unknown. The type 7 secretion system is used by mycobacteria, such as tuberculosis bacterium, the causative agent of tuberculosis, to infect host cells. So mycobacteria that do not express the type 7 secretion system, when they are engulfed by host cells, they end up in a compartment called the phagosome, where they are broken down. Tuberculosis, on the other hand, is engulfed by these host cells, by these immune cells, and end up in a phagosome, but there it can survive and even escape from this phagosome with the help of the type 7 secretion system. There it can then multiply and further infect the host. So why would it be of interest to determine the structure of secretion systems, or any protein complex or macromolecular complex? So proteins derived from genes fold into three-dimensional structures, which to a large extent determine their characteristics and how they are able to perform their functions within the cell. All domains of life use proteins in their cells and other macromolecules, and without proteins and other macromolecules, life would not be possible. So what is the traditional way to obtain structures of proteins? So the traditional way is to isolate the protein from its natural context. So it's typically overexpressed in a cell. The cell is then broken up. And then biochemical properties of the proteins of interest are used to isolate it from among the other proteins. Then with different structural methods, it can be structurally characterized. One of these methods is single particle cryo-electron microscopy. So the isolated protein is placed on a sample carrier, rapidly frozen, and individual particles of this protein complex are imaged. Then computationally, all these individual images of the protein complex can be combined to form a three-dimensional structure of the protein. But isolating proteins does come with downsides. First of all, isolating a protein can be difficult, and isolating a protein carries the risk of altering its structure or losing binding partners. Also, by obtaining a structure within the cell, you get structure in relation to its environment where it performs its function, so it's easier to link function to its protein structure. And also other protein complexes that you are not initially interested in may be present in the cell. So in order to determine the structure of the type 3 and type 7 secretion system during intercellular infection of human immune cells, we first establish a cryo-electron tomography workflow. So the first step of this workflow is to rapidly freeze the cells to preserve the fine ultrastructure of the cell and to protect the cell against the harsh environment of the electron microscope, the vacuum particularly. After freezing the cells, the problem is that most parts of the mammalian cell are much too thick to directly image with a cryo-electron microscope. so we need a method to thin them down. In order to accomplish this, we use a focused ion beam to prepare what's called a lamella, a thin window of only 200 nanometers in the cell, which is prepared by removing all the material above and below a section of the cell at a shallow angle. And this 200 nanometer thin window is then thin enough to directly image with a cryo-electron microscope. After preparing these thin windows, the sample is transferred to a cryo-electron microscope, where we can record overviews of these lamella to find features of interest, in this case, bacteria during intercellular infection of immune cells. And there we can acquire what's called a tilt series, which is a series of images recorded at different tilt angles. The reason why we acquire multiple images at different angles is that we can then reconstruct them into a three-dimensional volume containing the area of interest and the protein complexes of interest. Here is an example of a two-dimensional slice from such a three-dimensional volume. In the red box, you can see many different protein complexes and other macromolecules in here, among which is the type 3 secretion system in the red box over here. And then to further increase the resolution, it's possible to extract many hundreds or thousands of these cubes containing particles of interest, and then iteratively align them using computational methods, creating a new reference, and then iteratively realign everything to create a three-dimensional structure of a protein of interest. This is what we did for the type 3 secretion system, and this is the structure that we were able to obtain. What we were also able to resolve here is the cytosolic portion, the lower portion of the complex, which could not be resolved previously, or was not resolved previously for this species. And because we obtained the structure within its native cellular environment, we were also able to see how these complexes interact with the host cell. So in this example here, there's two type 3 secretion systems where the needle, the syringe part of the complex directly interacts with the host membrane, shown in red here, and where the host membrane is deformed by this interaction. Next, we acquired a similar data set with the aim of obtaining the structure of the type 7 secretion system in mycobacteria, also during intercellular infection of human immune cells. However, we were not able to actually find our type 7 secretion system. There could be a number of reasons for this. One of the possible reasons that we were unable to find it is the difference in contrast if the bacterial envelope of mycobacteria compared to other species, like Yersinia enterocolytica, where the species in which we obtained the structure of the type 3 secretion system. So if you look on the image on the right, the contrast in the envelope is much darker compared to the contrast on the left for the bacterial envelope, the membranes. This does not mean that this data was useless, because we were able to see many other complexes in there, including a bacterial storage compartment called the encapsulin, marked here with the red circle. And we combined this data together with data from isolated protein complexes of encapsulin to determine structures of the encapsulin, the fully assembled encapsulin shell. And these data help us give insight into how these encapsulin shells may be able to assemble. So to summarize the thesis, we successfully implemented the cryo-electron tomography workflow, and we used this workflow to obtain the structure of the type 3 secretion system during intercellular infection. This provides insight into the structure in the native cellular environment. Next, we collected a large data set of mycobacteria with the aim of obtaining a structure of the type 7 secretion system. However, we were not able to actually find the complexes. But we were able to use this data to obtain, together with data obtained on isolated protein, to obtain structures of the mycobacterial encapsulin in different structural states, which gives insight into how these complexes may assemble from its basic components. Thank you for your time and attention, and I'll give the word back to the pro-rector.
Thank you very much for a clear presentation and a concise presentation. We have a few more minutes for opposition. And the opposition will be opened by the chair of the assessment committee, that's Professor Rötlingsberger, who's professor of biochemistry of apoptosis in the Department of Biochemistry in the University of Maastricht. And for today, he has also accepted the role of secretary of this committee, for which I'm very grateful.
Thank you, Mr. Pro-Rector. Dear candidate, I would want to start with giving you my compliments about the thesis, the work you've done. And these compliments also go to your promotion team. I'm fascinated by the work. I'm a biochemist, and we look at the biological system in tubes, etc. And we also look at the structures, as you also indicated in your presentation, but of isolated proteins. So the path to elucidate structures of proteins in their natural environment, that fascinates me. And I'm really excited by this path of research and what you accomplished with this thesis. My compliments. But not only compliments, I have a couple of questions. And in your presentation, you already touched upon one of the big quests you have, and that is to elucidate to visualize the structure of the T7 secretion system in the Mycobacteria, probably because it's so pathogenic and it can be used as a target for therapy. As you mentioned, up to now, you were not able to visualize that. And you mentioned that in your thesis in chapter 6 in the general discussion. But what was lacking for me to understand why you don't see it, and you mentioned one of the reasons you think you didn't see it, is that there is a protocol lacking. How did you infect the cells? Which cells did you infect? And how was your infection protocol?
Highly esteemed opponent, thank you for your kind words and your question. So the cells that I used are primary macrophages, and we isolated monocytes directly from human blood, which we differentiated into human macrophages. And then we infected these cells with Mycobacterium marnum, which is closely related species to Mycobacterium tuberculosis.
And they also have the T7 secretion system.
They express the type 7
and
secretion
also
system.
The ESX1.
They also express ESX1 specifically, okay. And they are known to require the type ESX1 for the translocation from the phagosome into the cytosol of the host cell. Mm-hmm. We saw translocation taking place. We saw bacteria that have translocated afterwards. So that means that ESX1 must have been expressed at least transiently at some point.
Yeah. But what was the time point that you take a look at the infected cells?
I tried different time points. I think the most common time point I used was 18 hours by heart. I don't recall. Okay. And I think I took a moment where roughly half the bacterium have translocated, roughly half of them have. And
how did you visualize the location in the cytosol, or how did you determine that these bacteria were in the cytosol?
You can see whether there's a phagosomal membrane around the bacteria or not in the tomograms.
Yeah. I'm also interested in this part. So I took a look into the literature, and there I got with old literature showing that Mycobacteria, but it were the Mycobacteria tuberculosis, that in vitro, when they infect macrophages or dendritic cells, differentiated into dendritic cells, they saw a latency period of 48 hours. So, and they assessed the translocation of the phagosome into the cytosol by the proliferation of the bacteria. So how do you reconcile these data with your location of bacteria in the cytosol?
I'm not familiar with this particular paper, but I think the fact that we can see translocation taking place, that we can see that there's no phagosome around the bacterium already shows that they have translocated and have escaped from the phagosome.
Yeah, and that's not an artifact. So what I'm trying to get clear is, what is your evidence or your strong notion that these bacteria have produced the ESX1 functionally?
So I think it's correct that it's indirect evidence, the fact that they have translocated, and the strong evidence that ESX1 is essential for this process. And it's true that we don't have the direct evidence that ESX1 is expressed, and additional experiments with.
So, would you think it's wise to take a look at a later time point, at 48 hours, or even longer, 72 hours, to take again a look at with your in situ?
I think the most sensible approach would be to try to get a confirmation that ESX1 is expressed maybe by the Western blot at different time points, possibly also extending the time points.
Yeah, okay. And one of your explanations is that the periplasmic space has a high density, so it's dark.
Yep.
And I thought in your thesis you also made an analogy with the T3SS, where you see this fine needle coming out of the bacterial wall and punching into the membrane of the phagosome. But that space is not electron dense, and you didn't see any needles. So what do you conclude from that? So let's assume there is ESX1 produced.
Yeah. So I did indeed not see a needle, which could indicate that the type 7 secretion system does not have a needle complex. This is still unknown whether it has one or not.
That's not known?
No, we do not know whether the type 7... So we only know now from isolated structures, the structure of the on the inner membrane of the bacterium. We do not know how bacteria are transported across the outer membrane.
Okay, so that's still to discover.
Yes.
Interesting. Mr. Prorector, do I have a couple of minutes?
Certainly.
Okay. I want to switch gears, and as a biochemist, we in our textbooks, the description of the cell is enveloped by a plasma membrane. And in the 1970s, two people, Singer and Nicholson, they described the structure of the plasma membrane as a continuous layer, bilayer of phospholipids with embedded proteins, and they call that the fluid mosaic model. And we are still attached to that model. But that's not part of my question now, but just to give you a hint why I come to this thought, is we find experimental results that cannot be explained by this fluid phase, this fluid mosaic model. Now, your technique may give an opportunity to, at least that is my interpretation, to really inspect the plasma membrane of cells in situ and to challenge this model. Now, do you think along this way? So when I ask you, is your technique of your cryo-electron tomography with FIB-lamellae suitable for exploring the plasma membrane, its real structure?
I think it would be possible to segment the membrane and then extract subboxes along the membrane, and then you should be able to classify out the membrane. That's a pretty typical thing to do. recently, somewhat by chance, got a nice class of a member of the bilayer of the membrane. I did not obtain different classes. My worry might be, I think it's an interesting hypothesis to try to find heterogeneous areas in membranes. The risk may be that rather than being a single phospholipid, it's a collection of phospholipids altogether.
Yeah.
So it might be too heterogeneous to ascribe it to a single class. But of course, there's supposed to be different patches that have different characteristics. So I think it's an interesting hypothesis. To my knowledge, I don't think anyone has published anything on this directly yet.
Okay.
And there's also computational methods to try a similar approach to try to find transmembrane protein complexes.
Yeah.
So that could also be of interest.
Wonderful. Now, I'm very satisfied with your answers. Thank you very much, and I give the word back to the prorector.
Thank you, Professor Rudlingsberger, for your position. Then we go to the following opponent, who's also a member of the assessment committee, and that is Professor Saxe. He is professor in structural biology in the Ernst Röska Center for Microscopy and Spectroscopy with Electrons, and that is in Jülich, Germany, and Düsseldorf. And I would like to thank you very much for coming all the way from Jülich and Düsseldorf.
Thank you, dear candidate. I also would like to compliment you on your work. It's been a pleasure to read your thesis, the high quality, great figures, and also the way you told the story. So I would, as you probably, it's not a surprise, most of my question would be targeted on the technical electron microscopy end. And one of the things I was very curious about your work on the fiducial, using fiducials by this particular endocytic uptake that you proposed. So I was wondering there on the technical note, when I look at table 1 on this chapter where you finally conclude that you improve the quality of the tomograms, and perhaps you can walk me a little bit through this once more. What kind of statistical criteria did you use in order to come to this conclusion?
Highly esteemed exponent, thank you for your kind words and your question. The methodology that we use is that we independently aligned tilt series using either the gold fiducials that we added to the cells using endocytosis, or used patch tracking, which does not use fixed fiducials, but rather uses cross correlations to align different areas of the tilt series to get a better three-dimensional reconstruction. What we then did is use the residual, which is a metric of the mean displacement of each either fiducial or virtual fiducial from the cross correlation. And we directly compared these values, and we saw improved alignment using gold fiducials. And additionally, we also directly compared the visual quality of the tomograms, and we observed a better reconstruction quality using gold fiducials.
Mm-hmm. So, and you for that purpose, you used the same software, just two different computational procedures in a way.
Yes,
so
that's
I guess
correct.
IMOD or...
It was both in IMOD, we use patch tracking in IMOD, and IMOD for fiducial-based tracking.
Mm-hmm. Yeah. Yes, I understand. Did you also think about removing computationally the gold fiducials in order to assess this? Why would that be maybe an interesting approach?
So the contrast from the gold fiducials also contributes to the success of the patch tracking because it adds contrast. So indeed, if you had computationally removed fiducials, that may have potentially made it more difficult for the patch tracking to accomplish, which the contrast from the gold fiducial is normally not present. So that it arguably could have been a more fair comparison between patch tracking and gold fiducials. However, the result is likely that you would get a worse result with patch tracking than we already obtained.
Mm-hmm. Yeah, that is a really good conclusion. Yes. So one of the other things on this particular method was so now you really showed this nicely for particular applications where endocytosis is involved, right? Because these are being taken up. So what if I were interested in processes that are happening in the ER, for example? Could you use this method as well? Any thoughts on this?
It will depend a little bit on the magnification that you use. The larger the field of view, the lower the magnification, the more chance that there will be some gold fiducials in there. There's no way to guarantee that every tilt series will have gold fiducials. So you may still would have to use a mixture of tracking of the gold fiducials. An alternative option is also to use any inhomogeneous depositions present on the top of the lamella, either from redeposition during the FIB milling or possibly from the platinum layer as an alternative. However, using these is not as straightforward and not as accurate as using the spherical gold fiducials, or fall back to patch tracking.
Mm-hmm. So the critical point is really this uptake. Would there be any means to target structures to other parts of the cell?
So we looked at the distribution throughout the cell, and I think you have a roughly 50% chance of having gold fiducials in your field of view, but of course it will depend for a cytosolic structure. For the nucleus, of course, there is no chance of having any fiducials. So whether this is a suitable method does depend on your question, your biological question.
Mm-hmm. Thank you. So I also would like to go into the work in the following chapter on the structure of the injectosome, which you presented. So how did you assess, or what was the resolution? How did you assess it?
The resolution was about 3.5 nanometer to 4 nanometers. How we assessed it is that we split the data set in 2 even parts, independently align them, and use a Fourier correlation to assess up to which resolution the 2 independent half sets match with the 0.143 criterion.
Mm-hmm. So I've recently come across publications where they showed work leading to resolution even in situ to 3.5 angstroms, so 0.35 nanometers. So why is this not possible in your case?
So I think the main limitation here is the number of particles. So we had, for the type 3 secretion system, we had 202 particles to work with. During my current work in the UK, we also obtained a ribosome structure at 4.9 angstrom resolution in situ, but the main difference there is having 15,000 particles, or actually 70,000 particles to start with. So I think to get a much higher resolution for the type 3 secretion system, the main contributing factor would be more particles in this case, and at some point also the processing pipeline that you use also makes a big contribution. So the more modern processing pipelines, such as warp, can refine the motion correction and the reconstruction of the tilt series locally during the reconstruction process, and that gives a very sizable increase in resolution, in particular once you pass roughly the 1 nanometer barrier.
Mm-hmm. Thank you. And then, finally, I guess, so one of the things now, I guess that's why this was really a very great challenge to address this particular injectosome. I guess it's a hard nut to crack. But for the future, what would you try to do better, in the sense if you could dream up a microscope according to your perfect settings in order to push it into a similar resolution range that you've just been discussing?
So in order to acquire more data, I think the most realistic route would be to use automated FIB-lamellae fabrication, which is now quite possible to get more lamella. And the tilt schemes that I used took about 40 minutes, whereas if you use beam shift to acquire multiple tilt series at each set of tilts, you can reduce the time to about 5 minutes. So using that, those 2 methods, I think it's much more realistic to acquire a much larger data set. So I acquired about 150 tilt series. With these methods, I think it should be it should be feasible to acquire about 1,000 tilt series in a reasonable time frame. That processing all this data is still going to be a very significant task, but that I think would be the most realistic way to acquire more particles, assuming you want to be limited by doing it in an in situ context. The other approach could be to make FIB-lamellae of bacteria directly, because then you would have many more secretion systems per lamella.
Mm-hmm. Yeah, thank you, dear candidate. I'm satisfied with the answers.
Thank you, Professor Saxe, for your position. Then we come to the following opponent, who is also a member of the assessment committee, and that's Dr. Barcena. She's working in the field of electron microscopy in the Department of Cell and Chemical Biology at the University of Leiden. And thank you for coming all the way from Leiden.
Thank you. Dear candidate, first of all, I also want to join the congratulations on this wonderful thesis, which I really enjoy reading quite a bit. And I think is a prototypical example of the power of this beautiful technique, and also a bit of the limitations that we all have experienced. And I think a good example is in chapter 5, where you study encapsulins in vivo, and using this in situ cryo-ET allow you to unravel, discover these tail-like extensions in the encapsulins that had never been visualized in vitro. I think that's a very powerful example. However, of course, the images are not enough to tell you which protein or proteins constitute these tails. So this is an open question. And in the discussion of this chapter, you said that further studies, insights within the cells are needed to reveal this point. So to me, it doesn't seem a case of just collecting more images. So I would like you to explain what you would like to do to address this question. What could you suggest to do, perhaps with imaging, but also with other techniques?
Highly esteemed opponent, thank you for your kind words and your question. So assuming we would want to proceed characterizing the nature of these stills, I think, so one of the difficulties may be that they have not been observed on isolated protein. Of course, the question then remains, is it a very transient structure? But the difficulty there is if we isolate the complex, it's going to be very difficult to tell whether we just lost it or potentially whether it's an artifact that we are looking at. Perhaps a feasible approach could be to cross-link first, and then try to isolate the encapsulin, and then to use mass spectrometry perhaps to try to identify if there's any other proteins or structures attached to the encapsulins. This is not really my area of expertise, but that might be a feasible route to approach. The imaging route could also be possible, but it's going to be very challenging because these are very flexible structures. So I think you would need a lot of data to be able to acquire a good structure of these tails on the encapsulin.
Yeah, I couldn't agree more that imaging is not. And I was actually thinking along the same lines. But then imagine that you find some candidates, there is a place of imaging, in situ imaging, to validate your findings, or do you think that only, I mean, how would you follow up with in situ cryo-ET to validate candidates?
A cryo-correlative microscopy could also potentially be of help. The density of but then you need at least a candidate for the tails to fluorescently label them and see if they correlate in their localization. In terms of the density of the encapsulins, that could also be a feasible approach potentially. But then the difficulty is finding a potential candidate for these tails.
I agree. That is the challenge. Okay. You could also then perhaps knock out when you should find a candidate, you could knock out.
Yes, that could also be a good approach. Yes.
Yeah. Okay. Well, another fascinating challenge for the future is this search for the type 7 secretion system, and I felt, it's really a challenging project. In the discussion, you seem to indicate that you collected a lot of data, and therefore it's very unlikely that you didn't see it. This has been addressed before. I would like to talk about it now from a bit of a different angle. You collected 200 TIL series, which is a lot, but at the end of the day, these are very high magnification images of only 100 nanometers, 200 nanometers lamella. So I was wondering if you, how much of the membrane, the total area of the membrane you sample, and whether, yeah, this is a significant percentage or not.
So I think, so we have about 200 nanometer thin lamella, roughly, or measured 185 in the type 3 secretion system paper. And I think we sampled a large amount of membrane also, especially considering that ESX5 is required for the bacteria to survive, and it must be constitutively expressed. So a risk factor is that ESX1 that is only transiently expressed. We don't know at this point. I think we have a large enough data set to say that it's unlikely that we can easily see it. You can never really exclude it, of course. That's the...
Yeah, of course you would agree, as was discussed before, that this depends on the copy numbers that you have still, right? Okay. So then you propose ways forward with cryo-correlative, for example. I think, yeah, fluorescence, perhaps as you did with in the chapter with Yersinia enterolytica, might help to establish how frequent you have this protein. I agree. But I also agree with you that this will not be the panacea to solve your problem, given the differences, the constraints in resolution. So what I was wondering is, what way forward do you see? Because it seems to me that even if you had an arrow, a molecular arrow telling you the complex is here, the high contrast in the periplasmic space will impede any high resolution or higher resolution structural analysis by tomography averaging. So do you think there would be a way to tackle this contrast problem?
No, that's a good point. It would make any subtomolar averaging a lot more difficult with the contrast. So one approach could be to try different mycobacterial species. Not all of them express ESX1, of course, so then you maybe would be more sensible to try to aim for ESX5 or ESX3. It's definitely a very, very challenging complex, especially in these somewhat early stages during the thesis project for applying these techniques. Yes.
So so you also mentioned mycolic acids, for example, long-chain mycolic acids that are the special composition of this membrane. Is there any way to try to change the composition of the membrane?
Very, very short answer, please.
I think some mutants may reduce the density. I don't know by heart which particular mutant would be a good choice for that.
Okay, thank you very much. I'm very satisfied.
Thank you, Dr. Bacena, for your position. Then we come to the following opponent, who's also a member of the assessment committee, and that's Professor Moroni. He's professor in biofabrication for regenerative medicine, the Merlin Institute in the University of Maastricht. Professor Moroni.
Thank you, Mr. Prorector. Dear candidate, I also would like to congratulate you and the promotion team for a very interesting piece of work. In fact, actually, where I really learned a lot. I come from a slightly different field, but that does not mean that actually, in many cases, we have a lot of interest to go more in depth into understanding the cellular biological phenomena that are happening when, in my case, we want to regenerate a specific piece of our body. So to break the ice more for me than for you, I'd like actually the help of one of your paraninf in reading proposition number 3.
Number?
3.
The pursuit of high-resolution structures creates the risk of ignoring more heterogeneous structure states, which may, biological, be highly relevant.
Thank you. So I came from the macro side. Maybe sometimes we go to micro, we love to go to nano. And for you, nano is the daily life, right, in science. And this proposition actually triggered me, because is there a way, in your opinion, that we can go into that middle stage where you can still look structurally at the nanoscale, but indeed with maybe other kind of analysis, optical analysis, still look at a higher scale so that you don't lose that heterogeneity?
Esteemed opponent, highly esteemed opponent, thank you for your kind words, your question. I fully agree that for in situ, being able to integrate the cellular environment with structural states is definitely where we need to go more, even as a field. My comment on the different structural states is also still very relevant for isolated protein, I think, as we may tend to go for the one high resolution structure and sort of ignore the other structures that may also be of very interest. But in particular, in a biological cellular environment, any protein, there's really no reason to say that any protein or macromolecule is a bad particle that should be excluded. So I think our current hierarchical processing pipeline is not necessarily that well suited for the in situ context, and having a better pipeline where we keep better track of all our particles and assign all our particles, not just say this is a bad particle,
okay,
we don't want it here.
No, clear, but maybe I also like to maybe take inspiration from one of the pictures that you had in the presentation, which also, by the way, I really liked a lot. You had this beautiful picture of the membrane with the receptor entering into the inner part of the cell. There, for example, I missed, I think, in the picture, but maybe I'm wrong, the cytoskeleton. But could you maybe connect such a high ultra level detail such as interaction to the receptor on the plasma side with what happens actually at the cytoskeletal level?
I think the best tool currently available to accomplish this, that would be the TIX, which is a software package that allows you to recreate the three-dimensional structures that you obtained into the volume space of the tomogram. So if you independently obtain a subtomogram average structure of your cytoskeletal element and any proteins that may link your cytoskeletal element to a membrane, and then segment your membranes, I think that would be currently the best approach to visualize such an interaction. And indeed, trying to integrate that kind of information is definitely one of the key advantages of looking at protein structures in situ.
All right, thank you. Thank you so much. That's nice to hear. Then actually it brings me to another proposition, which I will not ask Pranif to read, but that seems to be really central in your thesis, and that's the in situ application of cryo-electron microscopy with FIB. And then you say the future of structural biology is in situ, right? And if I understand the thesis, that I actually agree with you. Again, from my point of view, in situ means also something slightly different, where we look at in situ cell biological processes. I want to have a cell alive, and I want to see, how in situ the cell behave according to certain stimuli. In your thesis, you do analysis in situ, but cells are, you still have to prepare the sample, you have to fix the sample to cut it with FIB. So is there a way that you can actually apply this kind of technology tool set with a living cell? If yes, I'd like to hear how. If no, I'd like to hear what you think would be an alternative.
I think the problem is going to be the harsh environment of the electron microscope and having an aqueous sample. So basically your cell is going to evaporate and you're going to have nothing left. So you would have to keep, on the one hand, keep an aqueous environment for the cell, and your cell's still going to be live. There would be movement. I think it would be highly unlikely that this will be possible, at least in the near future.
Yeah, that is what I thought.
I think our, to some extent, assumption is that when we rapidly freeze the cells and we get amorphous ice, that we preserve the cell in a near-native state. Of course, we cannot truly assess if there's potentially some artifacts that do occur from this process. But I think so far there's quite a lot of evidence that it is a near-native state at the very least.
All right, thank you so much. I'm satisfied with the answer. I give the word back to the prorector.
Thank you very much, Professor Moroni, for your position. Then we come to the following opponent, and that is Dr. Kudryashev. He's online, and he's professor for in situ structural biology in the Max Delbrück Center for Molecular Medicine in the Helmholtz Association, and it is in Berlin, Germany. And I would like to thank you also for coming all the way from Berlin, albeit online.
Yeah, thank you. How is the connection? Okay, I guess we can hear me. Mr. Candidate, I had a great pleasure reading your dissertation, and it was an excellent combination of very interesting biology and high-end technology and data-driven problem-solving skills, problem-solving approaches as well. On that, I want to start with the addition of gold fiducials, which I found very useful because it's a typical problem in situ structural biology that without gold beads it's hard to perform tomographic reconstructions. And you've shown that in a large fraction of your cell you can add gold fiducials and perform tomographic reconstructions using them. You have published this work, so I wonder if there is anything that limits people from doing this, because other than you, I have never seen anyone else doing this. Please go ahead.
Hi, esteemed opponent. Thank you for your kind words and your question. Also, to my knowledge, no one has repeated this far, and to be honest, I don't use it currently myself. In principle, any cell that anositizes gold, which in principle are most cells, are able to do this. So in principle, there's no limitation for anyone working with mammalian cells, most mammalian cells, to do this. Maybe one of the reasons people don't do this is that it's yet another step to figure out to optimize during the already somewhat complicated workflow. And I think the alternatives for patch tracking are also getting better. Perhaps the need for gold fiducial based alignment is becoming less, and also with newer subtomography averaging pipelines, the need, so with WARP and similar data processing pipelines, it's possible to improve the alignment of the tilt series later on during the data processing, and that to some extent alleviates you from the burden of getting as good an alignment in the early stages. So all you really want is a good enough alignment to be able to find your particles of interest. So it may end up being a dead end. I don't know. It was definitely very helpful for the type 3 secretion system. I think we'll have to wait and see whether people will still rediscover this or rather choose to go for patch tracking approaches and use more modern pipelines to improve the tilt series alignment during the subtomography averaging processing.
Okay, thank you. I'm very satisfied with the answer, and this also answered my second question. So maybe we can move to the work on your type 3 secretion system, which most of it, a lot of it we did together, right? So with my participation. So it has been previously shown that, or suggested that upon the function of the type 3 secretion, the basal body that's between the membranes contracts. However, in your work, the contraction was not that pronounced. Could you maybe speculate on why this was not the case, and maybe the system that you had is superior to the previously used systems?
That's a very good question. Of course, a large difference here is the intracellular context. So it could be a reflection of the actual reflection of the biological state of the secretion system. Perhaps there are simply fewer contractions taking place. Another factor here to consider is the low number of particles. So I think with more particles, we could have potentially obtained more different states, and particularly more convincing states, because once we started classifying, the averages did clearly suffer compared to our random controls. But it's a very relevant question, one that would possibly be answered by being able to acquire a lot more data in an intercellular context, possibly compare it to the non-infection context.
Any other methods that could be used to answer or quantify the contraction of the basal body, perhaps not related to electron microscopy?
It might be possible with very high resolution fluorescence microscopy, but I think it would be quite challenging. It would be possible to get a lot of data, so that might save you there. Not sure if there's another method that you were alluding to, but that might be a good approach.
Mm-hmm. Okay, thank you. So my last question is that about the evolution of technology, and you have seen it firsthand. So previously people did cryosection, which was very difficult. Then you have done a lot of work with cryo-FIB milling, and now I see that you switched to plasma FIB. So is this the kind of the end game for the sample preparation for structural work? Or do you think still there can be some fundamental improvements on how we prepare sample for cryo-imaging?
Oh, a very good question. So in my limited experience with cryosections, I would say I prefer using FIB-lamellae, and I think the field seems to agree, as FIB milling seems to be the dominant technique. Having said that, FIB milling is not perfect. So in a current, so one of the downsides of FIB milling is that it creates a damaged layer to the surface. Our work is not yet published, but it's deposited in. So we show some evidence that this is indeed the case, and it affects the resolution you can obtain with subtomography averaging. Then another consideration is the stability of the lamella. So there tends to be some movement in some lamella not so much. So I don't by no means think that lamella are perfect. I don't really have the answer what would be the better method still to accomplish this. So I think it's the best we have for the time being, and I think we can already accomplish a lot with it, especially with the recent advances in cryo-electron microscopy, tomography, and the newer data processing pipeline. But of course, it is very much in the interest of the field to try to continue to find better ways to prepare better lamella, but also to find possible alternatives to lamella.
Thank you, Mr. Candidate. I'm very satisfied with the answers. And that's my turn to the next examiner.
Thank you, Dr. Kudryashev, for your position. And now we are coming to the following opponent, and it's Dr. Diepold. And he's working at the Department of Ecophysiology in the Max Planck Institute for Terrestrial Microbiology in Marburg. And thank you, of course, coming all the way from Marburg.
Thank you, Mr. Prorector. Also, thank you, Mr. Candidate, for that very nice thesis, which I enjoyed reading a lot. And I think it nicely highlights the power of cryo-ET in microbiology and also in infection biology, as you have shown. You've also shown some example of elusive structures. Type 7 ESX1 is one of them. But I was intrigued by these really striking structures that you identified, but could not kind of correlate with any known biological entities that you show in the last chapter. So what would be your way forward towards identifying these structures, especially keeping in mind the tremendous progress in these automated structure prediction algorithms?
Highly esteemed opponent, thank you for your kind question. So with the recent developments in AlphaFold, it is possible to create large libraries of unknown protein predicted structures of overall high fidelity. So assuming we are able to obtain a structure at a high enough resolution to allow docking of such unknown predicted structures into the newly obtained structures, that could be a very powerful way forward to simply screen a very large number of structures, look for cross correlations, how they may fit in there, and then try to find unknown protein, what is the identity of this unknown protein complex.
So what is missing? I mean, couldn't you kind of run AlphaFold or AlphaFold multimer over the whole proteome that you would expect to find in a cell, and then create these kind of structure predictions or outlines to scan your micrographs? What is missing? What are the next steps towards this goal?
So I think the libraries of these unknown proteins are being made as we speak at a very rapid pace. So in order to see whether it would fit, that would be in principle known algorithms for doing so. It's a cross correlation with different orientations and translations. So I don't know if anyone implemented a method to link that to any of the databases. I think the main limitation is actually to obtain is to obtain the structures at a high enough resolution in situ. Currently, this process is still quite manual. There are more methods coming to automate or semi-automate the particle picking, also for unknown proteins, but it's still a very time-consuming process. So if I look at how I spend my time, a small portion of it is actually spent on sample preparation and microscopy data acquisition or lamella preparation. The vast majority of my time is actually spent processing data and trying to get structures out. So I think if we really want to move the field forward, our efforts should really be on automating segmentation to find membranes, finding unknown proteins, and then more automated pipelines for obtaining structures within cells.
Okay. I mean, one intermediate workaround that you also discussed is CLEM, so correlative light electron microscopy. And I wasn't quite sure what was the main obstacle for the ESX1 in that case. I guess you have functional fusions. So is there any... What was the obstacle of using this method for the type 7, which of course was the most elusive? And maybe as a second part to the question, would there be any alternatives, electron-dense tags, for example?
So we did not have yet the fusion protein to actually do the experiment. So I think that could also be a very good step to try to still elucidate, to find the type 7 secretion system. And indeed, electron-dense affinity tags could also be an option. However, all the tags currently in existence are still quite imperfect. There's a lot of work being done at the moment. And one of the difficulties here is also that we don't know the outer membrane components. If we had already identified an outer membrane component, it would be a lot easier to use these electron-dense tags to tag it from the outside, because now we have to tag them from the inside, which is more challenging.
And maybe as a very short...
Thank you, Dr. Diepold, for your opposition. Mr. Berger, the time appointed for the defense of your thesis has passed, and the degree committee will now withdraw to discuss the quality of your thesis and of your defense. And I request that you and your company await the results of our deliberations and our return in this room. And having said that, I suspend the meeting.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes. In the meantime, let's take a closer look at Maastricht University's renowned teaching method, problem-based learning. Once the prorector reconvenes the session, we'll tune in and continue the live stream.
Problem-based learning, or PBL. What does that mean exactly? 3 of my fellow students and I will show you around. Every week, we analyze a different case or issue together. We discuss the case and everyone can contribute different perspectives to the group discussion. If we get stuck, our tutor helps us out and suggests what we could do next. I prefer going to the library to prepare. Here, I can focus and I have quick access to books or journals that help me understand the case. Today, I can also train my stitching skills in the Skills Lab, where you can immediately put into practice what you've learned. After a day like this, I like going to the gym to clear my head and get ready for the next day.
At UM, you meet people from all around the world. Hello guys. Some of them are doing their exchange semester here, and they often say that PBL helps them learn and retain things very easily. I can understand why. It's a very active way of learning because you have to bring your own perspective to real-life cases. You have a lot of freedom to manage your time, your studies, your hobbies, and your work. Of course, that also means a lot of responsibility. Right now, for example, I'm arranging my exchange semester in Madrid. How cool is that? In this group session, we're the managers who have to allocate the resources of a real company. This is how we put into practice what we learned this morning. Studying here means being proactive and learning to plan well. Prioritizing and performing well under stress are great skills that help you develop as a person. But now, it's time to grab a coffee at the Student Service Center.
Right now, we're at the Brightlands Camelot campus. Here we can apply knowledge from lectures and tutorials in a practical setting. This helps us understand what we have learned and further develop our lab skills. Today, we're determining the amounts of cholesterol in various products. What I really like is the project periods at the end of each semester, where we complete a full research project that includes planning, collecting data, analyzing, and presenting the findings. That way we learn how research works and we're able to see what it's like to be a real scientist. After practicals, I have to write a lab report that also helps me process everything I have learned today. UM has a lot of learning spaces where you can work on your own or with other students. This evening, I'm meeting my friends for a movie night organized by the MSP Study Association.
If you study law, you have to read quite a lot. Not all information is relevant, so you learn how to easily find the information you need to solve your case. In the afternoon, I have to give a presentation, so I like to practice it with a fellow student. Later today, a lawyer is giving a lecture. This will help us better understand the case we're working on. Speaking in front of a group is quite exciting the first time round, but you get to used to it quickly. And having to present helps you also to adopt knowledge better. What I like the most is that we sometimes get to enter a plea in front of a real judge. These moot courts are really exciting. Now it's time for drinks with friends. Studying is important, but so is relaxing once in a while.
I reopen this academic ceremony. Mr. Berger, the degree committee here present, on-site and online, has assessed the quality of your thesis and of your defense. And in view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of Doctor. Professor Peters is authorized to confer upon you this academic distinction in accordance with Dutch university custom. And I invite your supervisor to now take the floor.
Please all rise.
Do you promise to work in accordance with the principles of scientific integrity at all times? Do be careful and honest, transparent, independent, and responsible.
I promise.
By the authority vested in us by law, and in conformity with the decision of the committee here present, I hereby confer upon you, Casper Berger, the degree of Doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. And after this official event, I have the permission from the pro-rector to give a little laudatio. And the laudatio is to praise the candidate for his academic achievement during the period that he was in our laboratory, but also during the defense. Casper is about 35 years old, and before I continue to praise him, I like to praise the parents by delivering such a good boy on the planet. He did really a good job, and I was wondering whether you would expect him to deliver such a nice presentation today, but you can be very proud. In 2006, you started at the university in Utrecht, where I also studied myself in the infection and immunity, where I also did my master's degree. And then in 2011, you graduated in the infection and immunity with my colleague Marka Woben on the tetra spanners in the exocytic vesicles, which I also did myself during my PhD for a little bit. So we have things in common. And Marka told me that you really did a good job during your master program. So we were very eager to recruit you. And thanks to Carmen and Rimo, we convinced you to come to Maastricht and join our team. And Casper started in the most horrible time for a PhD candidate, I should say, because the lab was not even open, and we were getting instruments into our laboratory. And a PhD candidate usually jumps on a train that is already running and starts doing experiments. And Casper was exposed for maybe 2 years with lots of difficulties in the instrumentation and getting things up and running. And so I really felt very bad many times. But you got help from other people, Andreas and Michael, in the setting up of the experiments that we wanted to do. You tried to work on flash and Rias and fluorescence microscopy together with Fritjof, and that didn't work very well. But I saw in notes just yesterday that already in 2018 you made a plan for the things that are seen now in the PhD, and most of the items that were discussed then are now beautifully explained in your thesis. You worked also on the faceplate, but didn't talk about that at all. Here, I don't think it was any success like for many others in the field, but you also worked on jet vitrification. And that has not been discussed, not in your paper as such, but in principle, you already had a beautiful paper more or less ready. And I feel a bit sad that it is not yet out there. And you should be part of a publication when one of your paranymph is picking up this particular subject, because you did exciting preliminary data that gave us so much promise that we got a grant and pushed it further. And now we have even an instrument that is about ready to make 5 prototypes. They go all over the world, and from there, we will bring it to our startup company, and hopefully they will sell hundreds of them worldwide. But you were at the basis, together with Frank Naples, who is also in the audience, and some of your colleagues. You went also in 2018 to the tomography meeting, and there you did great work. And you presented a great talk in Groningen, where I felt like, wow, Casper is getting really an academic scientist of high level. So you, in the time when you developed, as we already highlighted it just when we worked together, you developed yourself scientifically tremendously. And I would like to, in my last 3 minutes, highlight 3 of your transferable skills, which you will transfer to your next job, which is in Oxford, in Yorkshire. Oxford, actually, near the beamline, where you work in the great institute and continue your work. So we discovered, Casper, that you have a skill in earning trust and creating confidence among people. And that is a really important skill, I think. And as I mentioned already in the beginning, you are showing extreme perseverance in the face of adversity. You manage to stay on board and not to leave us despite all the difficulties in the beginning, and you really push it to the end. We saw today that you can convey your vision, driving a point home. And we have learned, and we saw today also in your beautiful defense, that you are a critical thinker. And critical thinking is not so trivial. And there are not enough of those in the academic level, but you are one. And then, to a big surprise, which was not to me so obvious in the first 2 years, you are an extremely good writer. And we haven't discussed it yet, but chapter 2 is in a beautiful review about the work that Casper has been doing. And he already was working on it in 2018. And he wanted to submit the paper by the end of 2018. It was submitted 2 years ago, and we resubmitted it last year. And only 1 week ago, we heard from Nature Methods that the paper was accepted. And Nature Methods is, in life science, the highest impact journal where you can deliver a beautiful review. So you really can be very proud. That will be cited a lot as well. And we saw today you are a very precise speaker. So with that, I think I highlighted all the elements that I know that are in you and that are very characteristic of you, and that will bring you to better, higher, more interesting jobs in the future, for sure, Casper. I'd like at the end also to thank the committee who evaluated your thesis and also were here today. In particular also Michael from Berlin and Andreas came all the way from Germany, but also you, Monse from Leiden, and Carsten from Jülich. And then the local committee members were also here. And I'd like to also thank the team who is with us, who really helped you during the PhD and also in difficult times in the beginning. I also know that people in Yorkshire are watching this event, and I know that your new supervisor is also there, Michael Greike, and I wish him good luck with Casper. You are getting a really good scientist, and I know already that you have one paper under revision in Nature Communications, so I see you are flying very well. At the end, I would like to say thanks also to the 2 paranymph. These are your nearest colleagues in the last 2 years. I think Carmen and Remo and I are very proud of what you have delivered, and also today. And with that, I'd like to close.
Thank you, Professor Peters, for a very nice and kind laudation. And Dr. Berger, also on behalf of the Maastricht University and the deans, I congratulate you with the honor you have acquired. I've seen a beautiful academic discussion, which is very good. And with my congratulations, I also congratulate Professor Peters, Professor Ravedi, Dr. Iglesias, and of course your partner and family, and friends and family also in the UK. I would like to thank the assessment committee for their work, of course, and Lou Peters, Joshua Felu, and the Pedel who made this all technical possible. And I hope you can celebrate this day today. There's a long day ahead, so there's time for food and drinks, I think. But not in our building. You're going to another venue in Maastricht, I've heard. So before I close this meeting, I have a few announcements. We are going to stay here for a picture with our online opponent, and then on the stairs a picture. I can ask the people here present to leave already the room, and you can already go to the venue where the reception is, so that we have time for the picture. And then the candidate will follow you soon. That's the idea. Of course, the photographer has to stay. Yeah? Having said it all, I wish you a very nice day, and I close the meeting.
Klein beetje verder naar voren.